Good morning, everyone, and thank you for the one who actually made it so early. It's really kind of you. um, I'm going to try to present a bit of the work that we're doing in my growing group research that I hope will grow bigger in the next few years. And I will talk mainly about the different approach that we use, but they mainly use mouse and human data and try to find a new disease modifier and new target for ALS and FTLD. So I'm not going to spend too much time on it because I'm not a specialist and I'm not a clinician, far from being that. But as you know, ALS is mostly based on diagnosis of clinical symptoms. I mean, also other biological parameters, but it's based on upper and lower motor neuron involvement in this disease. But you know that there is also this growing evidence that show that basically multiple causes could cause ALS and lead to similar clinical pictures. I would call ALS more as a syndrome that all lead to UMN and LMN involvement. And what's interesting is that it could explain why some people have pure ALS and pure motor neuron involvement, while others will develop cognitive and behavioral impairment as well, with some people meeting the criteria for full FTD. So what we know in this clinical continuum is that somehow there is a shared pathology in the people who have ALS, the one who have been diagnosed with ALS and FTLD, and the one who are in between, so they have motor neuron disease with some kind of cognitive and behavioral impairment as well. But we still don't fully understand. We know that TDP-43 is involved in most of the cases, where it aggregates in 97% of the cases. We all know this famous figure from Ling et al. In 2013. But we need to better understand the role of TDP-43, but also FUS as well in these mechanisms. And so in my talk today, I'm going to try to split it in three different parts. The first part, I will talk about ALS irritability. I will ask the question of whether ALS is genetic. Of course, I think we all have the answer. It's partly genetics. In the second part, I will focus on how we can use cross-species transcriptomic to help us identify genetic factor and protective factor and new therapeutic targets eventually. And in the third part, I will switch gears and show you that we can use the same kind of approach to find disease modifier of the phenotypes. So when I talk about ALS and genetic, I like to picture this as an iceberg, basically, because what we see right now, and it's been shown yesterday remarkably, is that we know that the genetic cases that we have right now, they explain a third of the cases, but we have 2/3 that are missing. So we are only on the tip of the iceberg, and we have all the other genes that might be involved. It could be other coding variants, intronic variants that has been shown yesterday. It's also interesting, but we know that it's only 1/3 of the patient for now, and yet ALS irritability is actually extremely high. So from really old study from Amar Shalabi, performed in twin individuals, you basically show that the genetic component in susceptibility is explained 60% of the variance. So 60% of the variance in susceptibility is explained by genetics, with the rest being other things. It could be environmental as well. So I first asked the question of, can we use the genetics to actually pinpoint a specific molecular determinant of the disease, right? So we start from the genetic architecture of ALS, and we try to integrate it with multi-omic data and try to see whether this genetic risk factor are enriched in specific pathway and specific cell types. And that's what we did. So we actually use a really simple approach that is called linkage disequilibrium score regression here that you see as a really dot plot. It's super easy to read. So we basically have all the individual diseases here that we tested with Alzheimer's disease and ALS here. And when you take the GWAS from an Alzheimer's disease patient versus control, and you basically looking at whether it's enriched in specific cell types, what comes out is microglia. And no surprise, there is a huge deal on microglia and Alzheimer's. When you do the same thing for ALS, what you see coming out are basically neurons, and inhibitory and excitatory neurons mostly. And what is also interesting, and what I found really interesting, is that if you look at some neuropsychiatric disorders, especially schizophrenia, you see that inhibitory and excitatory neurons are also there. What is actually really cool is that there is this study that was shown a couple of years ago from the group of Jan, who actually showed that there is a strong genetic correlation between schizophrenia and ALS. And there is another study which actually showed that you have an increased risk of psychotic episodes in C9 or 72 kydrates, which is actually really interesting and actually match up with the data here. So what does it tell us? It tells us that here in the genetic architecture of ALS is increased, the risk is increased in region, in genes which are highly enriched in excitatory neurons. So we have the cell types now. So now can we use this information and the new technology to actually try to understand what's happening in those excitatory neurons and try to find new potential targets? So that's what we did here. We basically took advantage of single-cell RNA-seq from human data, was being published in 2024 and also in 2021 on the motor cortex of healthy individuals from back in Italy. And so at the same time, we use clean longitudinal data sets from mouse data with SOD1 mutation, and we purified here, cortical spinal neurons using retrograde labeling, and we basically sampled this neuron at different time points, okay, using FACS sorting. What we did then is that we integrated both data sets, and we tried to see whether the cortical spinal neuron projection signatures is enriched in subtypes in human motor cortex. And so what you see here is a UMAP plot. You see basically inhibitory neurons here, excitatory neurons are right there. Here you have all the glial cells. And what you see here on this side, that the MCSN signatures is highly enriched in a really tiny subpopulation right there that it's called L5ET. So the L5ET are basically layer 5 extra telencephalic neurons. So basically what we call long projection neurons that go from the cortex to the spinal cord. And they're basically, if you look at the data, you can actually realize that it's only a tiny proportion of excitatory neurons. It represents in total less than 0.5% of excitatory neurons. So it's really, really small. And those are the ones that actually degenerate in the mouse, but also partly in humans, even if it's they're not the same cell exactly. Not the same cell exactly. So I'm going to pass all through the detail of the analysis, but basically when we did this, we actually prioritized two cell types that were really affected in mouse and humans, and we turned out to the L5ET neurons, and we basically performed a cross-species weighted gene co-expression and network analysis. So basically, it's an unbiased way of looking at whether genes are co-regulated in a specific manner in mouse and humans, and whether it's conserved or not, okay? And then we kind of prioritized master regulator candidates using different approaches here that I will not develop for now, but you will see it here. So when we do this, you see this really nice heat map where you have the cell types at the top with the different excitatory neurons, inhibitory neurons, and microglia. And here you have the master regulator that were prioritized by using our approach of cross-species WGCNA. And so what really was really interesting that you observe actually two clusters. One cluster of master regulator that are upregulated in excitatory neurons and one that were not. And among them, we actually identify one gene that is called CREB3, which show also a high signal in genetics, okay? Which means that by integrating all of this, whether it's transcriptomic and expression and human genetics, we actually prioritize the single genes that seems to be upregulated in cortical spinal neurons, also in excitatory neurons, but seems to be also strongly enriched in genetic signal. So we move forward with this, and then we look at the genetic itself. So we had access to a cohort of whole genome sequencing data from patient and controls, and we did a burden test, so rare variant burden analysis. So what we do here is that we're basically taking rare variants, we're aggregating them over genes, and then we're looking at which gene is showing strong enrichment in rare variants. And so no surprise, what comes out as being significant, SOD1, TBK1, they're actually known as gene, but one that actually comes out as being significant is actually CREB3. And you actually see here the locus map plot of the CREB3 signal. It's actually a rare variant that is significant here. It's an arginine 119 glycine, and it's present in 0.4% of the population. And now it's really interesting in that it seems to be decreasing the risk of having ALS. So you have basically compared to KIF5A, UNC13A, and C9orf, you have a 40% reduction in the risk of developing ALS when you carry this rare variant. So we actually move forward, and we try to look at more functional data, try to look at whether CREB3 activity is changing in patients, in the brain, in the blood, and whether it correlates with clinical outcomes. So what we do here, we did here, is that we use brain and blood data. We look at the CREB3, what we call the CREB3 regular, which means that basically CREB3 direct target would carry the motif in the TSS, but also the one that are actually distantly regulated by CREB3. And so when we look at the correlation of this CREB3 regular activity, we see that it's positively correlated with survival, whether it's in the brain and in the blood. Okay, so the more CREB3 regular activity you have in the blood or in the brain, the longer is the survival time. What about the variant itself? So it's actually becoming a bit more difficult because, as I said, it's more enriched in control than it is in patients. So you have less carrier in control than you have less carrier in patients than you have in control. So when you come to do survival analysis, you're actually losing a little bit of power, especially because you don't have a lot of ALS patients. But when we do this, we actually realize that the carrier of the variants is actually a slower rate of disease progression. So slow ALSFRS are a slope that you see here, quantify, and also increase survival time. What is also interesting is that when we look at the canonical target of CREB3, which is SEC24A, you see just a peak here in ChIP-seq. You see that when you clone it, and we basically clone and overexpress CREB3 and mutated CREB3 with a reporter, we found out that the mutation is actually increasing over 50% the expression of SEC24A, which seems to be a gain of function. So the mutation seems to be increasing the activity of CREB3 that seems to be leading to this potential protective mechanism, which is actually quite interesting. So what was also really cool is that in the burden analysis, we actually showed that it was not only a single variant that was associated, but multiple variants. But the other variants being so rare that you're losing power, especially with low sample size. So we actually try to increase the sample size and increase the number of control, and what we found out that we have other actually rare variants and ultra rare variants that seems to be depleted in patients and enriched in control. Some of them are actually being pursued now by Nathan, who is a PhD student in my lab, and he's trying to study the effect of R168H, which is actually decreasing by 80% the risk. So you have an even stronger effect because the variant is even rarer. And also pursuing the R155W, which seems to be also decreasing. It's also even rarer in the general population. So here you have just an illustration of where the variants are located. You have the R191G, which is not in the BZIP domain. So it's a BZIP protein. So basically what happened that it's anchored at the ER, and once you have a signal, it's being cleaved and it's being sent out into the nucleus, and it basically binds to DNA through this BZIP domain. Okay, and so R119G, which is the one we found, the first one we found is actually before the domain, but R168 is actually in the middle of the domain. And what you actually found really cool is that they're both arginine substitution. So it seems to be doing something to the protein that we don't quite understand. That's actually the job of Nathan. But just to mention that it's actually not isolated, and the group of Aaron Gittler in Stanford, they actually found the same thing in motor neurons. So when you did single-cell RNA-seq in motor neurons in the mouse SOD1 models, they found that the most resilient motor neurons, they overexpress CREB3 in the nucleus. So it's actually really cool because it's already been replicated in the completely independent labs. So that's what for the first part. So we can use mouse data and human data, integrate them, and try to find disease modifier. That's one example that we're actually pursuing, and that's the job of Nathan, as I said, was trying to understand more of how overexpressing CREB3 could protect motor neurons from degenerate. But you can use this approach in a total different manner and try to find disease modifier that would increase the risk of either phenotypes, whether it's ALS, whether FTLD, whether cognitive or behavioral impairments. And so in the second part of the talk, I will more focus on this approach that we use. And so I'm going to switch gear a bit, so I hope I don't lose everyone. But here I'm going to focus a bit more of ALS associated with first mutation. It's been really nicely shown yesterday, and I'm going to just give you a brief introduction of mutation in the first gene, which are responsible for a severe familial form of ALS, and it basically mislocalized into a cytoplasm. Okay, so the basic question that we ask is that could that FUS cytoplasmic mislocalization can drive cognitive and behavioral impairment. Okay, so in the lab, we have this really nice model where we have basically a truncation of the NLS domain that causes basically FUS to mislocalize in the cytoplasm. And that's a model that Raphael has developed in the lab. And so what we did in those animals, she induced the mutation only in excitatory neurons, okay? And so we performed single nuclear RNA-seq on those animals. Same approach than before. We had a single nuke from a mouse, we had a single nuke from a patient, and that we try to identify a common set of regulated genes. Okay. So we basically use the same approach than before. Instead of looking only at differentially expressed gene, we look at co-expression, which is a little bit more powerful because it doesn't rely on thresholding and p-values. Okay, it just tells you this gene is co-regulated in this gene is co-regulated in your first animals. Can you see the same thing in both models? Okay, that's a basic question we ask. So here's basically an illustration. What we did, we focus on the alpha AVT neurons, which are actually susceptibles in patients. We look at first delta NLS mice, which I just presented, okay, and we perform co-expression analysis and identify gene models. We then try to identify the one that were mouse-specific, the one that were human-specific, and the one that were conserved across species. Of course, we can argue, and we have been arguing a lot about whether we should be focusing on stuff which are human-specific and stuff which are mouse-specific or conserved. I like to believe that we've been working in animal model for quite some time now, and they have to be relevant in some way, and that if something happened in those models, it should be observed also in humans. So I try to, focus on conserved stuffs, and that's what I've been doing here. So here's an illustration of what a co-expression analysis looks like. You have the UMAP plot that show you basically all the different models that are associated and expressed in our models, okay? And what it tells you is that the more closer to each other are the point and the dot on this UMAP, the more co-regulated are the genes in these models, okay? And you see that, for instance, you're gonna have a really nice model in the mouse, but really sparse in human. When you look at this here, the turquoise one, you see that it's actually really, really dense in both species, okay? It's only qualitative. Of course, we need to quantify that. And the way we quantify it is that we look at how well the module is correlated to disease association, so ALS in our cases. And so we do it in mouse, we do it in humans, and we see that in most of the models, they actually were in opposite direction, except for this one, where we have the same direction of the decreased expression of the moles in mouse and humans. So we identify a set of specific genes that is downregulated in ALS patient and downregulated in our first knock-in mouse in excitatory neurons. Okay, it's cool to have a models, but it's cool for bioinformaticians to have a models. You know, what we want to look when you have a models is what do you have in these gene models? Is it important? Is it interesting? Okay, so we basically did basic gene ontology on the models, and what comes out is basically cilium organization, really important for ALS CRISP, we know this, through NEC1 and all the kind of genes that affect the cilium organization. Tubulin, RNA binding, and microtubule transcription. So really cool, interesting Go term that comes out of these models. So then we try to put some functional aspect on these models. Okay, we have a model of genes that represent roughly 600 genes. So is it related to any kind of RNA binding protein that's been previously associated with ALS? Okay, so the way we did this, where we approach this always in an unbiased way. So we look at single srRNA RNA-seq of 125 RNA binding protein in cells. Okay, so that we knock down RNA. We use data where we knock down RNA binding protein in 125 of them in cells. And so basically for each of these replicate, they basically perform RNA-seq, okay? And then we actually look at the expression of the regulon, okay? In all the 125 RNA binding protein which has been downregulated, okay? And what you see, and I rank them from 1 to 200, it's actually 200. Well, you see that the second one is actually TDP-43, okay? So we have a mouse. When you knock down TDP-43 in cells, the most downregulated is this mold, okay? It's related to TDP-43. So we have a model that seems to be TDP-43 dependent, okay? The eighth one is actually FUS. So in the top 10, we have FUS and TDP-43, okay? I didn't mention the other one, but they're also extremely interesting, especially, and some of them have been associated with ALS risk already. So we actually decided to confirm this, whether we observe a decrease in the expression of the models. When you knock down TDP-43 in iPSC-derived motor neurons, you have a strong decrease, no doubt about it. When you look at FTLD TDP-43 sorted nuclei, so basically nuclei in the frontal cortex where you have loss of TDP-43, you have a decreased expression of the models as well. So we have models that are actually dependent on FUS and TDP-43 strongly. So I'm going to pass on this, but basically, we have identified a set of commonly affected genes. We identify a regulon that seems to be dependent on TDP-43 and FUS, and it's affected in ALS. Is it affected in ALS vulnerable neurons? That's the next question we ask. Okay, so do we see a decrease in this regulon in ALS vulnerable neurons? So that's part of Rafael's work. She's a nice poster over there, and I think you'll all be happy to look at it. But she basically had the lead on all the mouse work. So basically what she did is that she mislocalized FUS in human excitatory neurons and basically behavioral characterized those animals to see whether they had behavioral-like symptoms of ALS and FTLD. I'm not going to talk about it here, but basically the bottom line here is that when you mislocalize FUS in the cytoplasm, you have cognitive and behavioral impairment, which is associated with a decrease of the RBP regulon. Okay? And then the next question we ask is that can the RBP or the FUS regulon can be an indicator of ALS-CI or ALS-BI? So if it's associated with cognitive impairment specifically. So we use here again an unbiased way of doing it, looking at individual patients and data that we have, where we have diagnosis of ALS, but also evaluation of cognitive function using the ECAS scale. Okay, so we have basically clustered the individual. Here you see ALS individual, and ALS individual classified with cognitive impairment, which means that they have an ECAS score that is below a significant threshold. I'm not going to talk about it because we can talk hours about this. But what you see here are the different domains that are evaluated by the ECAS, the verbal fluency, the memory, so it's visual spatial memory, so it's usually more affected in AD cases than it is in ALS-FTLD cases. The language and the executive function. So, and here you have the expression of the FUS and the RBP regulon, and here you have basically the TDP-43 burden as well. So when you look at this heat map and this clustering, what you see is that you have ALS-CI patient clustering over there, okay? And they all basically seem to be affected in all single domain, okay? And here. What you see here is that it's correlating really well with the decreased expression of the FUS and the RBP regulon. We actually quantify this. I'm not going to show you here, but if you want to look at the paper, you're more than welcome to go and read it. But we actually quantify it, and it's correlating really well with the cognitive impairment in patients. Okay, and that's basically what's highlighting here, is that individuals with ALS-CI have a decrease in the FUS regulon and the RBP regulon. Okay. Do we see it specifically, if we look at ALS and ALS-CI patient, do we see it decrease specifically in neurons that are vulnerable in those diseases? Okay, so we look at excitatory neurons because we already showed that it was mostly excitatory neurons, but you know excitatory neurons are a really large group of cells in the motor and the frontal cortex that could be split in, you know, upper layer, a deep layer, and this upper layer you have different type of subtypes. Okay, but you know it's been classified everywhere, so it's okay. I'm not going to go into detail about this. But what we basically observe is that the FUS regulon is actually significantly decreased in ALS-CI patient, even more than patient without cognitive impairment. That's what we basically show here. This is really fantastic work from Jenna Gregory. She actually performed FUS staining in patients with ALS and ALS-CI, and she basically showed that you have an increased FUS pathology in patients with ALS and cognitive impairment in those individuals. So, what's the bottom line? The bottom line is you have RBP regulon that is downregulated in L5-ET neurons and is mislocalized in ALS and CI patients. Now, if we come back to the single nuke data, look at controls, ALS and ALS-CI, what do we see to this L5-ET population? Do we see different in proportion? So does it mean that basically those individuals who have been classified as ALS-CI, could they meet actually the diagnosis of FTD because they have strong decrease, possibly, of L5-ET cells in the frontal cortex? And so that's what we quantify here. You have a control, the ALS and the ALSCI patient. This is basically the L5ET cells, and you see a significant decrease of the proportion of cells in L5ET. Here it's quantified in ALSCI patients specifically. Okay, so we have a specific downregulation of deep layer neurons in the frontal cortex in BF44 and 46 in ALSCI patients. So we have a models of genes. So we repeatedly show that at the transcriptome levels, we have consistent expression and downregulation that is related to TDP43 and FUS, but I'm really interested in, do we have an enrichment in these models of ALS genetic risk factor? That's a question that I always ask, is that can it be helpful for genetic discovery? And so I try to look in an unbiased way again, where I took a positive control, which is the ALS GWAS. I took negative control, so I randomly sample in the genome a set of genes, and I basically look for enrichment in genetic signals, okay? And so what we show is that for the first regulon, you have almost 100-fold enrichment in signal for ALS genetic risk factor, okay? Which is less than ALS GWAS, which is our positive control, but which is way more than just randomly sampling the genome. When you look at the ALS and FTD continuum as we speak, we do see that if you do overlap with the gene that's been associated with ALS, with an FTLD, and ALS and FTD, we see that the regulon overlap, the gene that overlap, they have been associated mostly with ALS and FTLD. Okay, so what we did is that we try to actually test whether in the RBP regulon we would find gene that could be specifically associated at the genetic level with ALS and cognitive impairment, okay? So we perform basically a GWAS. So we look at the first regulon and we look at GWAS, so whole genome sequencing data from the ANSWER ALS cohort. And we basically perform an association between the variants, which has a MAF above 5%, in each of the RBP regulon first genes, and we associate it with ALS-BI and ALS-CI. What we found out is that we have a really significant hit on the one gene in the RBP regulon, which is FBXO16. We did the same thing with ALS-CI, and what we found out is that HOX is actually quite significant in the association. WWOX, it might tell you nothing, but it's been recently associated in cognitive impairment in Parkinson's disease dementia. So it basically drives the transition for Parkinson's disease to Parkinson's disease dementia. It's been associated with cognitive score, mini mental state, MMS score in Alzheimer's patients. So it's actually really cool to find it out here. But FBXO16 seems to be specifically for behavioral score, okay? And so we actually dug a little bit deeper now that we have access to large biobank scale, and I'm asking the question of whether the RBP regulon is downregulated, okay? FBXO16 is downregulated. So what happened if a patient carry a protein truncating variant in FBXO16 in a general population? Does it mean something? Does it do something, okay? So we look at the unbiased way using the UK biobank data. We actually extracted the patient who carry PTV and FBXO16, which is a small proportion of the 460,000 individuals. And we perform an unbiased behavioral characterization. I'm not going to talk about it here. But we look at basically MRI. We look at structural and DTI MRI. Okay, and we extracted all the metrics from the DTI MRI to look for specific association. What we found out is that the carriers of the PTV, they have a significant decrease in the AFA in the gene of the corpus callosum, which is really truly affected an FTLD patient, but also in the incident fasciculus, which is actually a frontal lumbar fasciculus that comes out from the frontal and temporal area. So it's actually quite interesting. So they have structural abnormality at the DTI level, at diffusion MRI level, but also look at structural MRI. So we look at T1 data. We basically perform association between PTV and volume of the gray matter in frontal temporal area in the left and right hemisphere. And what we found out is that patient with the individual, because they're not patient, individual with a PTV loss of function, they have a decreased area on the left hemisphere and decreased volume on the left hemisphere, but not on the right one, which is also quite interesting, which means that FBXO16 PTV carriers show frontotemporal atrophy in BF46 specifically. So we look at different regions. We actually look parcellation of the cortex, like it's been shown yesterday, and we actually found out it's specifically to BF46, the same region that we actually study in our ALSCI patient with single nuc. At the end, you know, we wanted another potential proof that those individuals who carry this PTV may be at some risk of developing neurodegenerative disorders, potentially frontotemporal dementia, or ALS with frontotemporal dementia. So we look at plasmatic biomarker. And so when you do this with Alzheimer's, FTD, ALS, even not Parkinson, but vascular dementia as well, you do see that all those patients, they have an increased neurofilament level in the plasma. What is quite interesting, and has been shown recently repeatedly, is that individual who have diagnosis of dementia that have NFL that is increased, but also GFAP, okay? You have an increased GFAP in the plasma, okay? And so when we look at ALS patients, we have an increase in neurofilament, but no GFAP expression increase, which is actually cool because it means that when you have dementia associated with motor neuron disease, you have this dissociation between NFL and GFAP. In FBXO16 carriers, we have an increase significant in neurofilament compared to control, okay? And we also tend to have an increase in the expression of the GFAP. When we do a randomized permutation and we compare just the mean value, we actually do see that the GFAP is increased also, but not with the GLMLs. So what's the bottom line here? I mean, if I want to summarize, I guess, all this part is that for this part, we actually, again, we started from single nuke data from a mouse models that we have behaviorally characterized that seems to be replicating, behavioral like symptoms observed in FTD patients. And we compare it to human data and try to identify common, and we identify those disease modifier. And what I want to point out is that at the end, we started from the models. From these models, we're trying to characterize the function of these models, how it is regulated, basically. And then, in these models, we're trying to identify, can we pinpoint disease modifier? And that's what we did here. So you can do it to find disease modifier of phenotypes. Here's the case for FBXO16, but you can also try to find disease modifier that could protect from the disease or could decrease disease progression as well. For the one who are interested more in, there are a lot of data in this paper. I couldn't present anything, so if you're interested, you can just look it up. And so with this, and the most important part, I'm going to thank everyone because it's it's a collaborative effort before everything, especially Dupuy's lab. So Raphael, we did all the mouse work that I've not presented here. Nathan was doing all the work on the CREB3 part. We just been funded, so we're going to start doing cool stuff now because we've been struggling with money. So now we're going to try to address the problem real problem here. Luc, of course, all the people in the STEP lab, collaborators. So Emily, who have been collaborating with, she's in New York, and she's been really wonderful and helping us with correcting those ALS and LCI patients, providing with single nuclear RNA-seq data as well. Jenna Gogo, University of Aberdeen, was actually has been really helpful in this project, and also James Schroeder at UPenn, who helped us a little bit on the story. And also, of course, Albert Rudolph, Adrian Occhio, who are the clinician we're working with for the whole genome sequencing, and Brian, who have nothing here, but also will be part of this project. The funding, and I will take questions. I hope I wasn't too long.
Thank you, Salim. Very nice talk. This is open for discussion.
Salim, thank you. Thanks a lot. It was really, really interesting. And I just want to ask one question. We have also recently worked on multi-omics from human data. And one thing we realized is that we were really big sex-specific differences. So you were not talking about males and females here. Did you see any differences between males and females, or did you always look at it all together?
So if you're talking about, if you're talking from the single nuke perspective and transcriptomic, I'm always looking at all of it together. I'm not trying to make any differences. For now, I'm sure if I look into the data, I will see that there are huge differences between women and men, but I'm trying to actually find common ground between both sexes. But I'm sure there are differences, for sure. But I haven't looked at it.
Excellent talk, thank you. Do we, maybe I missed it, do we know what CREB3 does? And is there, have you looked at slow versus fast progressor to see if there is a difference?
That's a good question. We don't know what it does. Because if you look at CREB3 literature on neurons, there is nothing. So there's a lot of work to do now. And we haven't looked at fast and slow progression either. We didn't do this. The problem is that, as I said, since the frequency of the allele is higher in controlled ALS in ALS patient, you need to have a lot of ALS patient to look for to stratify the data. So we couldn't do it here, but it's something we could do in the future if we're gathering more patients, of course.
Yeah, great talk. Thank you very much. So how large is CREB3? So would it be suitable to be put in an AAV vector? Or what happens if you overexpress CREB3 in the end, therapeutically? I mean,
it's kind of big, so I don't think you would put it in AAV like this. But I don't know what happens if you overexpress it. What I can tell you is that the little bit of study that look at CREB3 and overexpression, it was mostly related to immune system. You know, and it seems to be really highly involved in replication of viruses. So, and that's why I'm also interested. I had a talk yesterday with I don't remember his name, but he presented the interferon work, and I think it has a really strong impact on the virus. But I don't know what is exactly the role of CREB3. We don't know yet, actually. So.